This is part two of your immune system notes, and we're on the back of the big sheet that we started last time. So this is what's called the acquired specific response. So this is a bodily response that, unlike what we talked about before, is going to improve the second time it encounters these antigens or the virus or the bacteria or whatever it was. It's going to learn from previous experience. And if it encounters the antigen ever again, its response is actually going to be much more swift and significant in subsequent infections. So this is really where the brunt of the immune system comes from. And there are two types of responses we're going to talk about. We'll start with the humoral response, and humoral just means liquid, like a humor, like the medieval humors, or just four different liquids. So humoral just means liquid, and this is really just going to mean the blood for us. So this is the bloodstream for us, and associate the humoral response with B cells. And what these B cells are going to do is they are going let me just move this up here. They are going to produce antibodies. Antibodies are just proteins. They're going to produce antibodies. One way to think of the immune system is like a military. Think of the humoral response as kind of being like the Air Force dropping chemicals. I know it's kind of a horrible analogy to think about, but in terms of kind of understanding how this thing works, I think it's actually a fairly good analogy. And then contrast this over here with the cellular response. So the cellular response is going, that's supposed to be an arrow. The cellular response is going to be T cells. And think of this as kind of like the army. Again, I'm not a huge fan of, killing, obviously, but army is kind of like man-to-man, person-on-person combat, which is kind of what T cells do. So again, the analogy, despite how bad it might seem, is actually pretty decent at getting the main picture across. And we're going to actually go back over here and start with the humoral responses and our B cells. So here's a picture of a B cell. And again, these B cells have, they have antibodies on them that we talked about last time. And these antibodies have very specific shapes, but they are activated by antigens. We'll get to helper T cells here in a second. They're activated by antigens. And remember that we talked about the bacteria last time and we drew two little triangular antigens on it. Remember, an antigen is just anything that the body doesn't quite recognize and treats as foreign. Instead of thinking about a bacteria now, let's just talk about COVID. Let's imagine that this is a virus rather than a bacteria. And basically what's going to happen is anytime the antigen on a virus or a bacteria attaches to the antibody on a B cell, it's going to activate that B cell. And when we first learned about B cells and studied them and took a census of them, basically, we realized that the body has over 1 million different types of B cells, which is a crazy amount. And what that means is that there are 1 million different varieties. There are 1 million different B cells that each have their own different antibody attached to them. And the antibodies have all of these different shapes due to very different, slight amino acid changes or different substitutions or things like that. And it turns out that every single antigen that we've ever encountered, that our B cells have ever encountered, has had a matching antibody already existing in at least one of our B cells. This is kind of the power of having so many, 1 million plus B cells, is that they've kind of covered all of the different variations, the physical shapes of antigens that have ever been encountered, which I think is really, really cool. So think of this, I'm going to go ahead and I'll say that there is, it's just, again, it's not like the B cells are planning all of these different shapes. It's just they generate so many differences in these antibodies that it's just kind of covered all the bases here. So I'll say random different shapes, and it turns out again all the antigens have matched at least one of the different antibodies, the shapes. And the body actually will destroy antibodies that match its own antigens. You know, if you don't want to think your own body's antigens are foreign and attack them. So your own shapes are destroyed. I'll say own shapes. Own shapes are destroyed. That way your body doesn't attack itself. Although it could, in which case you have an autoimmune disease. All right, so basically what happens is when a virus or a bacteria with these antigens attaches to an antibody on a B cell, and again, you're just going to have to get used to knowing that an antigen is the foreign object and the antibody is the receptor. It's just going to take practice. Once that contact is made, and again, this is just diffusion, the virus isn't looking for the B cell or vice versa necessarily. They're just kind of floating around in the bloodstream, and if they hit each other at the right spot, the active site of that antibody, then it's going to prompt a cellular response. That cellular response is to divide. It's called clonal selection because you're just making clones of the particular B cell that got activated, right? The particular B cell. So let's just say this little triangle antigen is our shape. So this is like a triangle B cell. That particular B cell is going to make thousands and thousands and thousands of copies of itself. And there are two basic types of those copies. One type of copy is called a memory B cell. This is a B cell that has antibodies that match the triangle antigen, and they stay with you forever, right? They stay with you forever. They circulate in your body for life, and they can divide into more plasma B cells and more memory B cells. All right, so let's say can divide into more plasma B cells and memory B cells. Right. The other type of B cell is the plasma B cell. So a plasma B cell is going to mass produce antibodies. You're like, well, where do these antibodies come from? Well, they're mass produced by these plasma B cells. Think of antibodies as kind of like chemical warfare, and this is kind of why I went with the Air Force response. If you just imagine like dropping a whole bunch of I guess in agriculture, if you drop pesticides everywhere and you hope it kills a pest somewhere down there in the crops, it's just kind of what these plasma cells, B cells do. They just mass produce and spray these antibodies all over the body, and then hopefully some of them encounter the antigen that they were made to encounter. Right. They can produce 2,000 plus antibodies per second. Think about all of the ATP and the energy and the transcription and the translation going on here. All the amino acids that are needed for this. It's quite a lot of antibodies that are being made here. So again, think of these as kind of chemical weapons that are on the lookout, so to speak, for these foreign antigens. Right. So again, they're going to bind to the specific antigen that's related to the B cell. Remember, this was a triangle B cell, so it's going to make triangle antigens, or sorry, triangle antibodies. See, I said the same thing too. It's easy to get them confused. And again, they're going to bind to the antigen, the foreign protein or whatever on the coat of a virus or on a bacteria, and it's going to destroy it or inactivate the organism with the antigen. So it can physically surround it and immobilize it, or it can recruit white blood cells. It can recruit macrophages that can devour it. All right, and actually I got ahead of myself. I never even told you what an antibody was and how they worked. So remember, this is the antibody. And they have a very specific shape that I want you to go ahead and draw. So we're going to draw one shape like that. We're going to draw another one like that. These are proteins, so each of these is a polypeptide. So it's one polypeptide, two polypeptide. Draw a third one there, and a fourth one there. So this protein has quaternary structure. It has quaternary structure with 4 different polypeptides in it. And these are held together by disulfide bridges. And the important part is the ends, these 2 ends right here, because that is where they have the specific shape. Remember the millions of different shapes that we talked about? That's where those shapes occur, and that's the site where your antigen, there's the antigen, is going to bind. All right, so that's the anatomy of an antibody, and yes, you do need to know it. All right, that is an antibody. And again, these are released into the bloodstream by these plasma B cells, and they're just circulating in your bloodstream and your lymph system, basically, as long as you have an infection, and a little bit afterwards. Most of the memory, your lifelong stuff, comes from these B cells over here. All right. Let's go back up here and talk about T cells. Right. By the way, I also forgot to say that the B cells and the T cells are the lymphoid stem cells, or they're derived from the lymphoid stem cells that we talked about in the last video. So just to connect the back and the front. So the other type of cell here is a T helper cell. And these are activated by APCs. Remember that APC is an antigen presenting cell. So on the other side of the big sheet, remember we drew the cell with the MHC little platform here, and then the antigen was sticking out on top of that, and I talked about how it was like parading around the cell, celebrating its conquest. That's the APC. So that is going to physically bind, the antigen on the APC physically attaches, that's the APC, to the antibody, and it's going to activate the T helper cell. So the only difference here is that the antigen is attached to an APC, whereas over here, the antigen was on the actual virus or the actual bacteria itself. All right, so an APC with its antigen attaches to the antibody on a T helper cell. It prompts it to do a very, very similar thing. It's going to do clonal selection. It's going to make copies of itself over and over and over again. Just like with B cells, you get 2 types. You get, well, you don't get a T plasma cell. You get a T helper cell, and you get a memory T helper cell. These memory T helper cells are, again, they are with you until you die. But the T helper cells are going to, oh, can I type please? Are you gonna let me type? There we go. They are going to activate B cells, and they are going to produce cytokines that stimulate cell division. So let's look at these 2 effects in turn. The activation of B cells, let me grab my marker here. Let's see, where do I want to go? Let's go back up here. That's this line right here. So here's the activation of B cells. Oh, that's not the right line. This is the cytokine. Okay, fine. We'll talk about the cytokines first. All right, they're going to stimulate B cell division via cytokines. Sure, let's start with the cytokines. So cytokines are signaling molecules. These are signaling molecules that when they bind to the B cells are going to cause the cell to undergo mitosis, right? They're going to cause the cell to divide. So I'll say these cytokines stimulate mitosis, but also just like the cytokines can stimulate B cells, cytokines can also recursively stimulate their own T cells, which is why this loop here is also here. So go ahead and just write cytokine there so you know why there's a loop. So cytokines can also activate T cells in which case it just causes more T cells to reproduce and divide and clone, and you get more T helper and more memory T helper cells. So basically, think of T helper cells as cheerleaders. They are encouraging the reproduction of B cells, and they encourage the reproduction of T cells. So they are kind of cheering on the rest of your immune system and telling them to divide, 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 make more, make more, make more. That's what these cytokines that the T helper cells are, in charge of. So we can think of these guys as cheerleaders of the immune system. Sure, why not? Cheerleaders of the immune. Yeah, cheerleaders of the immune system. Those are my quotation marks that look horrible. All right. The other thing that T helper cells can do, again, is they can actually activate the B cell directly. This is where I got sidetracked before with the cytokines, and I just went ahead and included that right there. Right, so again, the B cells can be activated by antigens that we talked about before, and by T helper cells that we just now talked about. All right, cytokines can also stimulate the production of these cytotoxic T cells. So cytotoxic T cells are T cells, and they kind of behave like natural killer cells, and we'll see how that works in a second. But when these cells are activated, they also do clonal selection. So this is a theme, right? They're going to do mitosis, they're going to divide. And just like the other types of cells, you will get memory cytotoxic cells that stay with you for life. The other types of cells, the cytotoxic T cells, also called CD8 cells, but you don't need to know that. These are the cells that act like natural killer cells. So their job, just like a natural killer cell, is to go around and check all the proteins that are attached to the cell membrane of your body cells and make sure that they belong to your body. But if they find a viral protein or a bacterial protein or an antigen, I should probably use the proper term. Antigens are usually proteins. Then they react just like a natural killer cell does, and they either demand that it conduct apoptosis, and again, it demands it using cell signaling. It doesn't yell at it. It's all cell signaling, or it perforates it. It has these perforin proteins that basically just make a giant hole in the membrane, and water can come in, and the cell's contents come out, and the cell dies, right? And again, the cells that usually are attacked by cytotoxic T cells are cells that are infected with a virus or cells that are cancerous. And remember, the downside to the cytotoxic T cells is that they only destroy and attack your own cells, right? So only own cells. So this means that if you have a bacterial infection, it's out of the realm of the cytotoxic T cell. It has to be a problem with your own cells. They either have a virus infecting them, and they have these kind of abnormal proteins on the surface, these antigens, or it's a similar thing, but because of cancer. They're not going to attack bacterial cells themselves. And you're like, well, how did the bacterial cells themselves get attacked? Well, that's what the antibodies do from your B cells. Remember, the antibodies clog up the antigen, and if the antigen is on a bacteria, then they basically shut down and pile on the bacteria. But the antibodies, no, that's what I meant to say. The antibodies also recruit white blood cells. So. All right. I keep getting ahead of myself. These are receptors. Let's just say that I did this before I talked about the memory and the cytotoxic T cells. I got ahead of myself. So again, just like the helper T cells, they also have these antibodies on them that detect antigens from APCs, from antigen presenting cells. So there's your APC. These are the same receptors, by the way. These antibodies here. These are the same receptors as the T helper cell. They're not different. Same as the T helper cell. And as we kind of just saw, again, these are much more kind of aggressive in your face, almost, you know, killer cell-like from the backside of the big sheet. So this is much more kind of a man-to-man, soldier-on-soldier fist fighting. That's why we kind of went with the army with this kind of analogy here. All right, so if you take a step back, we're going to kind of summarize this really quick. Again, this is your acquired specific response. This is a learned response that improves over time, and there are two major cell types. You have your B cells and you have your T cells, right? B cells are activated by antigens or by helper T cells, right? Your T cells are activated only by APCs, antigen presenting cells. So that's the difference, all right? Once they are activated, your B cells will divide. All of these will divide into some form of memory cell. That's a commonality between all of these types of cells. So I'm going to kind of ignore the memory cell for right now because the interesting part, I think, is the other option. If it's a B cell, your plasma B cells make a lot of antibodies. And a good way to remember this is B cells make antibodies, right? Your T helper cells produce cytokines and they can activate B cells directly. Remember, cytokines are like the cheerleaders of the immune system. They stimulate cell division of both T and B cells. The cytotoxic T cell basically acts like a natural killer cell. It looks for body cells that have foreign antigens that it doesn't recognize and either commands the cell via cell signaling to do apoptosis, to do cell suicide, or it loads the membrane of it up with perforins, which are just proteins that puncture the cell membrane and pop the cell and destroy the cell. So pay attention to the vocab. Try not to get your antibodies confused with your antigens. And I will see you for the next video.